Welcome back. In early step, we already discussed how precipitation reaction, precipitin curves are. What is precipitation reaction? How precipitin curve is formed. So in this section, we are discussing about the visible precipitin lines. So the precipitin curve is formed as a result of precipitation reaction that takes place in a aqueous solution or fluid. In that way, in same case of when the precipitation reaction that takes place in a gel, this will leads or formation of a visible precipitin line. When this reaction takes place in the fluid, it yields the precipitin curve on another, and if it just takes place in an agar matrix or a gel, it will lead to the formation of precipitin lines. Precipitation reaction can also form in a agar matrix, and this line is formed as a result of two reasons. That is, when this antigen and antibodies are incorporated in the agar and allowed to diffuse each other forms of visible precipitin lines, and on other hand, when the antibody is incorporated in the agar medium or agar matrix and the antigen is diffused and an antigen is added to this agar matrix, so this will allow us to diffuse with each other and forms a visible precipitin lines. So there's the interaction between the antigen and antibodies is by the diffusion between them in the agar, and there are two types of immunodiffusion reactions. They are radial immunodiffusion and double immunodiffusions. So both this takes place on a semi-solid medium, that is agar, and this will help us to determine the concentration of antibodies and antigens, and also help us to compare the antigen concentration, and sometimes also and other uses helps to determine the relative purity of the antigen preparation. Now coming to the radial immunodiffusion. Here the diffusion takes place in radial form, where the antibody is incorporated in the agar matrix and the antigen is added into the central well, where this agar contains a central well, where we can add the sample antigen, and then they are allowed to diffuse each other and allowed for the diffusion and the interaction. So we can see, so this is the radial immunodiffusion, or this is the central well with where we can add the antigen, and this is the agar matrix which is incorporated with the antibodies, full of antibodies. And after diffusion or interaction, after allowing for the diffusion, a zone of equivalence is formed where the concentration of antigen and antibodies in the is same. So as a result, a ring is formed around the well in the form of, as a result, a visible line, that is, here the precipitation is formed in the form established in the form of ring around the well. So that's why it is called radial immunodiffusion. And the area of the precipitin ring is proportional to the concentration of antigen we added, or sample antigen. And so by comparing the area of this precipitin ring with a standard curve, we'll get the concentration of antigen of our sample, or we can determine the concentration of the antigen sample. Then next method is double immunodiffusion. Double immunodiffusion means here the both antigen and antibodies are incorporated with the agar matrix, and they are allowed to diffuse. they are allowed. Sorry, both here both antigen and antibodies are added in a wells of agar. This is a schematic representation of double immunodiffusion. Right one, where one side the well contains antibodies, and the other side it contains a well with the antigen. So, and it is by adding the antigen and antibody to this respective wells, it diffused through this agar matrix and forms a visible line at the area of equivalence or region of equivalence. A precipitation was formed at the area of equivalence. So this region contains equal amount of antigen and antibody diffused towards each other. And so here in double immunodiffusion, a visible precipitin line is formed, whereas in radial immunodiffusion it is in the form of ring of precipitation. And and this is established based on the concentration gradient, and equivalence is reached the visible line of precipitation is formed, that is we already discussed. Now this these are the difference between, from these two pictures we can easily understand the difference between both radial and immunodiffusion. In radial immunodiffusion, we are only the antibodies incorporated in the agar gel and we are adding antigen into the well, whereas in the double immunodiffusion, both the antigen and antibodies are added in separate wells on an agar matrix. Antigen on the one side and antibody on the other side. Antibody is in blue color and antigen is in the red color. Both are added in separate wells on an agar matrix and allowed to diffuse to each other and forms an a precipitin line or a precipitation in the form of a line, visible precipitin line. This line is called as zone of equivalence, where there is concentration of antibody and antigen is equal. In case of radial immunodiffusion, a ring of precipitation is formed around the well, where the proportion of antigen and antibody is in equal concentration.